So I'm going to present some data on genome editing in human pluripotent stem cells. I apologize in advance, my talk is a bit sketchy and wavy because I'm not going to present a polished studies, but many database and results based on our experience, but also experience of the BRC hPSC core facility, which has been created in Cambridge by the NIHR and the Biomedical Research Center, and I've been in fact working since 2009. And the goal of this facility is to derive hPS. So we derive probably thousands of hPS line over the years. And the past 5 years, we also started to generate basically hPS using genome editing. And we done more than 50 gene either knockout or gene correction for this modeling. And this BRC hPS core facility is also doing a lot of training. So we train around 10 PhD postdoc every year. We are involved in different courses, and we do help a lot of different other projects by providing consulting affiliation and also helping with grant. And really the key activity of this BRC hPS core facility is not only to derive the cell lines or to produce the genome editing lines, but really to help people to develop their disease modeling in vitro, or to develop their cell-based therapy approach. So what I'm trying to say is that this core facility not only generate the lines, but also look at the phenotype and help people to interpret their data, so which gave us an overview of the process. And so I'm going to go in different points on the genome editing, because I thought that would be useful to discuss this different point after after the panel. And the first point I'm going just to describe really quickly is guide RNA efficacy. And I think Cathy, I've already mentioned that, but it's a key aspect of the genome editing, which is often not mentioned, but clearly determine how good are going to be your knockout and how good are going to be your lines. And that's also really important in the interpretation of a lot of data that are currently published, showing that CRISPR-Cas9 could be toxic or problematic. Of course, that will be, this technology really depends on this aspect. And just to show you quickly how we have analyzed that in the past. So again, Cathy have presented that we use an inducible system that allows, in fact, to we constitutively overexpress Cas9 through a knock-in in the AVS1 locus with a safe harbor, which allows us to have a homogeneous expression of Cas9, but not too high. And then we basically knock in on the other allele an inducible guide RNA based on the H1 promoter, TETO. So basically, that means that we don't use a very high level of expression of Cas9 or a very high level of expression of guide RNA. And that just shows you the expression of reporter gene in safe harbor, and showing that our system is very efficient to express the different transgene in different cell type, and especially homogeneously in pluripotent cells. So that's the design of our system. So you have the inducible short guide RNA here and the overexpression Cas9 here. And that's the way we produce the cells. We target the AVS1 locus using zinc finger, not Cas9 in this case. And then after, we basically induce the activity and the expression of the guide RNA with tetracycline. And so to test this system, what we did is to basically target a transgene this time. And for that, we targeted another safe harbor, which is the ROSA26 locus. And we overexpressed the EGFP, the destabilized version of it. And then we design a guide RNA in silico and start to target this transgene basically. And so that's now show you the inducible knockout. And showing that basically in absence of tetracycline, you don't have any background, and that if you add tetracycline, you start to see the disappearance of the expression of the GFP. I think what is interesting here is that here you have basically a time frame, and that show you that basically the induction of the guide RNA can take a lot of time to basically target your loci. Okay, so here it took us almost 30 days to get rid of the expression of the GFP. So you need a lot of expression of the guide RNA to get an efficient targeting. So based on that, what we decided to do is to increase the number of guide RNA to see if we, by increasing the quantity of guide RNA, we could speed up the knockout. And that's what happens. It's just showing you here the basically knockout after a week, and you can see that basically very quickly, if you increase the number of guide RNA, you accelerate the knockout. So really, the efficacy and quantity of guide RNA you're going to transfect in your cells is essential. And then, of course, we tested the different guide RNA. So that's shown here. That just showing you that that's our guide RNA 1. And of course, now we have been a bit lucky because that's the worst one that we could use. And basically, this one was very efficient, but guide 2 and especially guide 3, we were able to basically knock out the expression of the GFP after only 2 days. But even there, I think what is important to underline is that even with the best guide RNA we can find, we never seen a full knockout more than 24, 72 hours after induction. The effect of the guide RNA takes a lot of time to act, because if you compare that to knockdown, a knockdown in 12 to 24 hours, you'll have knocked down your gene. With the knockout, it's more time. And Katie have mentioned that is because your guide RNA sometimes need to target several times the loss site you want to delay it to be efficient. And so, no, that's what we did also with Katie in collaboration. We did the same thing with OCT4. And again, when you look at OCT4 knockout, you see that the knockout take a lot of time to basically be very efficient. And that means that when you do this kind of inducible system, you get an heterogeneous population that are knockout and not knockout at the same time. So that's a key drawback of this kind of inducible CRISPR system. But so what I just wanted to conclude on that is that based on the experience, really CRISPR-Cas9 really depend on the guide RNA you have designed. You need to be extremely careful with that. And again, that's why it's very difficult to conclude anything on off-target, everything on toxicity, because basically we need to be sure that we're using the same guide RNA that have been optimized and designed in the proper conditions. And the basic rules to design a guide RNA are a bit magical, because you discuss with different people, they will give you different recipe how to design the best guide RNA. And that's something that we need to go a bit more in detail and probably to have standard how to design those guide RNA. The other aspect that we have encountered as a key challenge for the targeting in the BRC hPSC facility has been the targeting strategy. Because we end up sometimes with a customer that come with genes like MES1, for example, which contain certain isoforms. So you need to design a guide RNA that target one exon in there, and it's basically impossible. Okay? So on these genes, you can't do a proper knockout because basically you can't find a guide RNA that will knock out all the isoforms. You have also this example, which is WTAP, and in this case, WTAP is in fact included in another gene, which is SOD2. So that means when you design your knockout, you need to be absolutely sure you're not going to design SOD2 at the same time. And that's a bit of a challenge, I can tell you. We spent a lot of time trying to design the right guide RNA. And of course, now you have the usual standard deletion. So that's a GATA6, key gene for the differentiation. But when you do the knockout of the first exon, which is the usual strategy that everybody used to do CRISPR-Cas9, what happens is that you activate a secondary promoter, which is after this first exon, and you get a nice production of a truncated protein, almost systematically. And 4 labs have done the knockout of GATA6, 4 labs have basically got the same problem and published the same data. So what I'm trying to say basically, from past experience, this problem of truncated protein, isoform expressed, is a massive issue. 50% of the gene we have targeted, we get this problem. Okay? And especially with human gene, for a reason we don't understand, there is a lot of rescue mechanism that allow the gene to be expressed or partially expressed, even if you decrease one exon. And often people mention the mRNA decay as key mechanism. It's something we don't systematically see. So it's absolutely essential that you know well the gene you are targeting, that you really look at the expression of the protein. Otherwise, it starts to be very difficult to conclude if you have a full knockout. And that's I think, in my opinion, a more important problem than almost anything else. Then you have the Cas9 off target. So of course, that's something that everybody is flagging as a key issue. And so, for example, again with WTAP, we were asked to look at off-target effects. So we designed a lot of guide RNA, triggered RNA because of the difficulty we had with the genes. So we did a lot of targeting, and we generated a lot of clones. And then we picked 7 of those clones and did what Katie explained previously, is that we basically sequenced all the 21 potential off-target sites, and we never seen an off-target effect of those 2 guide RNA. And we used 7 lines, and we couldn't see any off-target. So we haven't seen an off-target problem with this approach. That doesn't mean that doesn't exist, but at least with the technology that most labs can use, it's difficult to detect. And so that's a key challenge because, of course, there is a lot of systematic genome-wide studies on those off-target effects. It's very complicated to perform because you need to perform that with the same guide RNA is going to vary between loci and gene you're going to target. It's going to be variable from different cell type. So to generate a general consensus on off-target, I think is going to be very challenging. And so what technology you're going to use to detect those off-targets is going to be even more problematic. So now the solution we advise to our customer most of the time is to basically use 2 sets of guide RNA, and then compare the phenotype you're going to get. But of course, that's implied that you end up with almost 12 lines per knockout or knock-in, because basically you need 3 cell lines, wild type, HETs, HOMs, homozygotes, 2 sets of guide RNA, so that's 6 lines, and you want 2 genetic background because you want to avoid any problem of variability. So you end up with 12 lines for knockout. Now, where is the time where we could do 1 mouse knockout lines and be happy? So that's where the field is going right now. Genetic stability. So of course, we test our line after genetic stability. We do this chromosome karyotyping because that's easy. We have a service in the hospital that does that at a cost, but does that very efficiently. And basically, that's 90% of the lines we tested this way, and most of our lines, 95%, will be normal. I mean, will not show any major genetic anomalies after CRISPR-Cas9. So for us, not a major source of problem. We do a bit of also CGHRA and this kind of things, and of course, then we see more anomalies because we have a deeper resolution, and 20 to 30% of the lines will be occurring deletion or addition of sequence. But what is important is that before we started this whole knockout pipeline, we spent a lot of time to find the cell line that will be resistant to those genetic anomalies. So now we did a massive exercise on, I think at the time, 20 cell lines from the hPSC project, and we did a lot of subcloning, targeting, long-term culture to basically detect lines that will be resistant to genetic anomalies. So what I'm trying to say is that, of course, now we have a very low level of genetic anomalies in your culture is because basically we select the line that are the most resistant to this process. And of course, on top of that, we did an exercise of differentiation, especially toward hepatocyte and pancreas, because that's the main focus on my lab. And we selected again the lines that have the highest capacity of differentiation to do that. So at the end, we end up with cell lines that are perfect for what we want to do, but may not be representative to all the situations possible. And I think a lot of labs actually are doing that. As we were mentioning before, for example, the pipeline of knockout at Sanger is done on a line called COF2, which is being selected very carefully for its resistance to genetic anomalies and its capacity of differentiation. So, of course, in terms of genetic stability, we get absolutely scared when Florian publishes paper and the other paper are published because p53 mutation, and we saw that each time we do CRISPR-Cas9 in our lines, we're going to end up with a mutation in p53, and that's why we can do CRISPR-Cas9. And then that was the panic, and then we decided to sequence a lot of lines. So that's just an example where we did 5 lines, multiple clones, and we sequenced the DNA binding region of Florian, but of course now what it shows is that we need to sequence all the other genes, so we're in trouble. But what we show here is that basically when we look at p53, we didn't see, or we never saw actually accumulation of p53 mutation in our CRISPR-Cas9 targeted lines, which I think means that basically if you use a classic conventional way of doing the knockout with a short transition expression of Cas9 and not a very high level of guide RNA expression, you probably don't have a lot of toxicity and p53 selection. At least that's our interpretation of what we do. So that's my conclusion of CRISPR-Cas9 genetic stability. Just know that we don't see, CRISPR-Cas9 by itself is not an issue. I think it's more the cell culture, again, it's more the cell line you use, and you need to control that, but that's very easy to do. Of course, we'll have a whole discussion about how much analysis we can do, or we can interpret the accumulation of those genome-wide mutation. Then the last things I would like to mention is variability between sublines, and that's something we started to discuss. I mean, we have discussed in the previous session. I won't go too much that. But of course, when we do targeting, what we do is that we subclone population. And if you have any heterogeneity in this population, you're going to end up by growing a subclone which may have different characteristics. And do we see that? We do see that time to time. For example, no, that's again a targeting, and that's done in one of our hPSC lines. And what you see here is that this clone here have a much better capacity to differentiate into hepatocytes. So it does occur that time to time. It's not the general, no, that's 3 other clones, so they are fairly homogeneous. But this clone in particular seems to be better to do differentiation for reasons we don't understand. And basically, what we do now, and that's of course what mouse CSL people used to do in the past, and that we turn us to try to subclone our human iPS to get working on an already subcloned cell line. But that's of course something that needs to be further characterized, and mean that basically you need to use several lines to characterize your phenotype, and that one line is not enough. So that increases this 12 number even further if you go into that. Okay, so what I just want to conclude that CRISPR-Cas9 is super efficient, but that's dependent on the guide RNA. Really, we need to do an effort on the guide RNA selection and how we define that. You need to know your gene well, and you need really to validate the knockout at the protein level. Off target, that's the big wolf, but at the moment we haven't seen many of those, but that may change with genome-wide analysis, at least if we do that systematically. Genetic stability, again, we don't see as an issue, but we'll see a change when we do genome-wide, as Florian mentioned. But again, how we're going to functionally interpret that is a very big problem. And variability between subline, it needs to be systematically addressed. And again, probably the only way right now to bypass that is to do more lines, unfortunately. It would be great to have a subset of super lines that we can use that have been characterized, that we can really basically use in the field, and that will be selected on their capacity to targeting, resistant to genetic anomalies, capacity of differentiation, but also potential for distribution and so on, and IP aspect. Okay, and just to thank especially Anne-Sophie from the BR, Anne-Sophie Lennartz, which is the manager of the core facility. We've done a big part of the work I've just shown today. Thank you very much for your attention.